This is the second recording for the cell unit in biology at Limestone Community High School with Mrs. Stout. In this section, we're going to talk about the types of microscopes that can be used really to look at anything small, but of course the cell itself, different types of cells, and the different parts of the cells. The image that you see on the screen is of a typical compound light microscope, and the functions of each part include the eyepiece, and it's pretty much what it sounds like. You actually look through the top to see the specimen, and the eyepiece usually has a magnification of 10 times. Now this is called a compound microscope because remember it uses a couple of different lenses together to show the image of whatever it is that you're looking through that's very small. Extending down from the eyepiece, you have the tube or the body tube, and that has a series of mirrors in it. And what those mirrors do is reflect the image back and forth from where the image is on the stage up to the eyepiece. So when you're actually looking at the image, it's going to appear upside down and backwards from what it really is. At the bottom of the tube is going to be the nose piece, and the nose piece is what's going to have the different objectives, and the objectives are going to have more lenses in them that will help you see in conjunction with the eyepiece lens at different magnifications. So you might have a high-powered, mid-powered, or low-powered objective, and you always start on the lowest power objective and work your way up from there. To figure out what the total magnification is of whatever it is that you're looking at, you take the magnification that would be printed on the objective itself and multiply that times the eyepiece. So let's say, for example, that you're looking at a specimen with a low power objective, and on the low power itself it says 4x, so that's 4 times what would be with the naked eye. So your total magnification would be your low power 4 times your eyepiece, which I told you at the beginning was 10, for a total magnification of 40 times what it would be like looking at with the naked eye. Let's say you then moved it to high power, and let's say that the high power had a magnification of 40 times on its own. So your total magnification there would be 40 times the eyepiece, which was 10, for a total magnification of what? That's right, 400 times what it would be like looking at through the naked eye. When you are focusing the specimen, you will, like I said, you'll start with the low power objective, and then you'll use the coarse focus to find it in place. And the coarse focus, or the coarse adjustment wheel, it's the same. It's going to be the larger of the two adjustment wheels, and it's called coarse because it is kind of a rough adjustment. It does move the body tube up and down quite significantly. You can actually see it. And then you would use the fine focus to kind of fine tune what it is that you're looking at to show it in greater detail. Something to notice is that the coarse focus adjustment wheel should only ever be used with low power. It should never be used with high power. Extending down, and something that this diagram did not label, this part right here is called the arm. And the arm elevates the body tube and adjustment wheels and the eyepiece up off of the stage. So this is called, this base here is called the stage as it's labeled. And on the stage are going to be 2 stage clips, and their job is to hold the slide in place. In the middle of the stage there's going to be an opening, which is called the diaphragm, and that's what allows light to go through. Now in this microscope it is going to be one that uses a mirror to reflect light up through the diaphragm so that it then goes to the objectives. There are some microscopes you can use that can plug in and they have like an actual light bulb here at the bottom. Neither, in my opinion, is better than the other. It's just, do you have electricity or not? There are also sometimes microscopes that underneath this stage and with the diaphragm opening, they will have an aperture wheel, and it will have different sizes of openings that allow different amounts of light through that diaphragm. So when we're talking or when we're using a compound light microscope, remember compound means many or together. So we're going to have many lenses. And of course we're going to use light. What a compound light microscope is going to do is just magnify the image itself. It's just going to make whatever you're looking at bigger. And it's going to use more than one lens to do so. For our purposes, what we would basically use in this class is pretty much only ever going to be a compound light microscope. The next one is going to be a transmission electron microscope, which can be abbreviated TEM. And if you look at the parts of these words, it can help you figure out what is being used. Transmission, to transmit, to go through. Electrons, so it's going to be one of the subatomic particles, one of the negatively charged subatomic particles. And with a transmission electron microscope, it aims a beam of electrons all the way through whatever it is that you're looking at. And it shows very fine internal detail. With the transmission electron microscope, this is how we can know some things like, rough endoplasmic reticulum, or rough ER, has ribosomes, whereas smooth endoplasmic reticulum does not. No ribosomes. A scanning electron microscope works very, very similar, so we're going to have the beam of electrons, but when you're scanning something, you're just passing over the surface of it. So think about like when you're scanning reading for class. You're not really getting into the fine details of the reading, you're just getting the overview, the surface. So a scanning electron microscope, abbreviated SEM, it moves a beam of electrons over the surface of the object to show surface detail and gives a 3D effect. A scanning electron microscope is how we can see things like the membrane proteins on a cell. I have never used an electron microscope in all of my super nerd classes that I even took in college. They are very, very particular. In fact, labs that have electron microscopes are usually on the ground level, if not below, because even on a third or fourth floor building, there's too much movement as people walk around, or even sometimes as the wind is blowing, and it will disrupt that beam of electrons and you won't get a very clear view or image. So for your test, the questions that will be asked will be something about like the use of the different microscopes, what is it you're trying to find, and then be able to tell what type of microscope that would be used. With using microscopes, you should be able to identify the two different types, the two different basic cell types, and every single cell ever discovered is going to be one of these two basic cell types. Prokaryotic cells, so pro means before, or not, or against. And karyo is a Latin word that means center or nucleus. So your prokaryotic cells are going to be more primitive. They do not have internal structures surrounded by membranes, so they don't have a true nucleus. They don't have true membrane-bound organelles. Your bacterial cells are going to be ones that are prokaryotic, so only bacteria. That is a question that you will hear now through May, and you will need to know the difference of the two basic cell types. It is one of the basic pieces of knowledge that you need to take from this entire course. Eukaryotic cells. So again, you can still see that it's got that Latin base of karyo, which still means center or nucleus, but now we have the prefix eu, and eu means true, complete, real. So eukaryotic cells are those that do have a nucleus. They do have internal membrane-bound structures called organelles. Organelles just means small organs, small pieces that have different jobs. In addition to the structure of the nucleus, there's a significant difference in size for your prokaryotic and your eukaryotic cells. Your prokaryotic cells can be from 0.1 to 10 micrometers. A micrometer is 1/1,000,000th of a meter. So you can see how small that is. And your eukaryotic cells can be from 10 to 100 micrometers depending on what type of cell it is. And that is the end of lesson 2.